Remember my graph where we nominate a compound, we get through the TPP and we nominate our compound? I can't tell you how many times I get the person coming in, I just nominated my first compound. We made it through our TPP. It's time to go to the FDA for a pre-IND meeting. I have to say, this is like a huge pet peeve of mine. I'm sorry for anybody I'm insulting. And the reason is because you don't have enough data to go to the agency to ask any decent questions. And so so I was just pointing out, you've made it through this point, you've nominated, but you don't have much. So what are you going to go to the agency and tell them? You're going to tell them you have a new product? That doesn't help you. You need to get information back out of them. So here's Michelle's general rules for the goals of a new chemical entity or a new molecular entity, if it's a biologic, on what you want to get out of a pre-IND meeting. So from a tox perspective, you generally want to have enough, you want to confirm the doses and the plans for your pivotal tox program. If you have an unusual biologic, there are ways you can get into the agency in advance of your pre-IND meeting with a pre-IND meeting and get some early feedback on how your tox program works so you know what to do. And then you can go in for the pre-IND. That's not the case for small molecules. It's just too well established. So for tox, you want to try to understand and make sure that your safety program is going to support your clinical program. You want to make sure that from a CMC perspective that the specs that you're setting up and the methods, and let's say you identify a potential genotoxic impurity, the way you're handling that and where you're setting your thresholds are correct. And then you really want to get feedback on your phase 1 program and early feedback on your phase 2 program where you're going to go into patients. So from my perspective, your prerequisites are the complete DMPK package, which is done because you already did your TPP. That's fine. But you actually want to go in and do dose range finding toxicology with TK and histopathology. You really, really need to find your dose limiting tox before you go into the FDA. Otherwise, you're going into them with nothing. They have no idea where your tox limit's going to be, so they have no idea how to help you identify your doses. And if you need to add any sort of special things into your tox program. From CMC perspective, I always feel like it's important to get through the process development, get through an engineering batch so that you can support the specs that you want to use, both for drug substance and for drug product. And then from a clinical perspective, you want to really have a solid synopsis for your phase 1 program so that you get solid feedback. So if you go in with nothing, you will get nothing out. And so my big play is, seriously people, you only get one chance at a pre-IND meeting. So please don't blow it if you don't have real data to go in and get responses from. And I've had this happen on more than one occasions where you go in too early, I get some package of pre-IND back, and it's just they've thrown the book at you, essentially like, check this guidance, check this guidance, check this guidance, check this guidance. You got no feedback because you didn't able to give them any data to respond to. And you won't get a face-to-face, you'll just get a written letter with just, here's the guidances to consider. So my plea is that we finish our TPP work, get our nomination. We actually do this level of work where we do a proof of concept study, we get some CMC, early CMC work done, and then we get our protocol kind of worked out. We know what indication we want to do. That's hard, much, much harder than you would think it would be to actually nail down what we want to do clinically for at least the first couple studies. And then we go into the pre-IND meeting. Then when you get out of that pre-IND meeting, you have the information from the agency to enable you to go into what are the most expensive studies that you will do. Your GMP manufacturing is going to be expensive. You know, all of these GLP studies are going to be expensive. And so it's a way to sort of get enough data to mitigate your risk when you go into the large spend. So you will have investors and people pressure you, and you're going to have to try to hold back on that pressure. That's not always the case. So I was talking about a new chemical entity or a new molecular entity. Pre-IND meetings for repurposed drugs where they sort of have established CMC or sort of an established profile, that's totally not the case. For an established product that you're just repurposing to a new indication, go in ASAP. I'm not going to get in your way. I'm not going to be a pain in the neck about that. So, because there you're just trying to get feedback on your clinical program, make sure it makes sense. One of the wonderful things about working with small companies is they're always looking for creative strategies to save money, always. And rightfully so. It's a hard area to work in because you've got great ideas, but you have no funding. And so you're trying to get whatever proof of concept data you can clinically to get an attractive investors. So I've got lots of folks that are always looking for unusual and different ways to get clinical data. So more than a number of times, I've had people come in and tell me, I'm going to do an investigator-sponsored IND because you don't have to do the tox or the CMC package. So that is 100% false. That is not how it works. So let me just quickly explain to you the differences between all the different IND options that you have. There are great reasons to do a physician-sponsored IND, but not always, you're not going to get a win on it. So you have the investigator or physician-sponsored, it's the same thing, IND. This is a research IND. These are typically sponsored by an investigator at MGH who has an idea to do something. It's got one site. You need all of the support, but I would say most of these types of INDs are repurposing drugs because they have some idea that using a weak antagonist of this will work in this indication. And so they open up a physician-sponsored IND to test that hypothesis. And it's very research-oriented. So you rarely do these in healthy, normal individuals. Emergency INDs, you can imagine what that is. It's sort of like the Ebola type thing. I mean, it doesn't happen very often. I've never dealt with one. The treatment IND is the one that you guys are most familiar with because this is the one that is going to end with an NDA, right? This is where you're going to collect the data and end with a marketing application. So you have some sort of commercial sponsor. You have multiple sites. You have your full package. And you usually start studies in your healthy volunteers. But then there's another option, and I'm not sure if everybody knows about the exploratory IND. You may have heard about this, but don't really know what it is. It's essentially for phase 0 studies. So in this case, you can get away with a much reduced IND package because you're going in to your tox, into your clinical study with sub-efficacious doses. And so typically, you'll do a phase 0 study to do like a distribution study or to do a little bit of PK, maybe. Just you don't need high doses for that. So if that was valuable, and it's not that valuable to most people, but if it was valuable to you, you could get in and have a more reduced IND package if you wanted to go into a phase 0 or an exploratory IND. All right, is there an IND loophole? This was just sort of a silly thing to remind you that the whole point of the IND is there is a law that says that you cannot ship drugs across state lines, right? And what the IND is the exemption to the law. So truthfully told, that if you were able to make your drug, manufacture your drug, and run your clinical studies and whatnot in the same state, you would not need an IND. So the question is that a loophole? It probably is because you wouldn't need an IND. But here's the thing, you still need an IRB. So an IRB is not going to let you go into, that's why we have sort of a duplicative approach to going into humans. You have the regulatory authorities and then you have the local groups that are sort of patient-centered. So while it is sort of a loophole, it's not really a loophole because the IRB is never going to let you go forward. So I tried to win you something for doing something cheaper, but it didn't work. Compassionate use, I don't know if you guys have heard of that. You may have heard about compassionate use. It's the same thing as the expanded access program. It's a named patient. It means that it's sort of a separate entity of a type of protocol that you can run where you're actually not collecting clinical data in support of a submission. It's just to get a certain patient on a drug. I can go into that later if anybody's interested in that. What I did want to tell you was just how can you use some of these options to create value with your small companies. And so that's the great thing about working with small companies is you get to see their very creative strategies. And so one of my companies did an exploratory IND. She has a peptide, and this is pretty amazing what she did. Generated labeled peptide, and I'm not kidding, she spent, you're going to die, she spent like $15,000 for GMP labeled peptide. And then she went in to did, I think, maybe 3 patients at the university and got early distribution data. And now this is for a cancer. So she was able to show that her peptide went straight to the cancer, lit it up, and lit nothing else up. And so here she is. She spent maybe a total of $150,000 on this whole thing and has human data on lighting up her cancer. So it doesn't have any efficacy data. She's showing that it gets there and it appears to be selective. So I think it's pretty high value for the cost that it took her to get that far. Here's an interesting one, expanded access. Remember, these are These are protocols that are not going to lead, they're sort of like physician-sponsored IND, but they're not really research-oriented. They're just to get a drug into an individual. GW Pharma, they have a compound, actually I think it's a cannabinoid actually, and they had it for some indication, and it doesn't matter what it was, and they ended up opening, or having these physicians open up maybe 10, 100 physician-sponsored INDs across a bunch of different indications. And so they got a bunch of anecdotal data across all these different indications on what their product was good for. And so then they found out it was good for this rare disease, and they opened up a treatment IND and moved forward with that. So I thought it was a pretty creative and low-cost way to get a lot of information. It's anecdotal, I know that, but it's enough to do hypothesis generating, and then they went and did their prospective studies. So here's our investigator-sponsored IND. This one I think is pretty good. A lot of people have a great idea. They have a new target that they want to explore for a new indication, and it's just awfully expensive to sort of build out, everything. So if you can find a mimic or a doppelganger or something along those lines for proof of concept in humans, and so if you want to target this GPCR, and you know that this guy is a weak antagonist, he's on the market for something else, you can actually open up an investigator-sponsored IND, working with an investigator, and test your hypothesis using the mimic. If that hypothesis comes through and you see the effect you're looking for, then you can come behind it with your fresh new chemical entity, your new IP, composition of matter, and come back behind it. So you can see what the package of data you're coming to a larger investor would be, which is, I've got proof of concept with an analog, and I've got my new chemical entity. Type of thing. So that's just a way to manage that.
This is a good one. What do you mean I have to make the drug before the IND? You really want to do that. Mostly because within the IND package there is certain CMC related information that you have to include, and you can't include it if you haven't already made the drug. Formulation process development. How are you going to make it? What master cell line, if you're using a master cell line, are you going to use? Has that been fully characterized? What about a working cell line? Has that been characterized? What is the synthesis route? Going back to the API and drug product purity, you have to characterize this. What are the impurities going to be? What are the specifications going to be? How are you going to know that the drug that the API or the drug product you just made is the same as the last batch that you made? And is the product going to be stable after a month? I think sometimes you could probably get away with adding the 1 month stability for the drug product after you've submitted the IND, but you do need the 1 month stability for the API. What starting materials are you using? How are those characterized? You need to provide the C of A's for those to make sure those are up to snuff. And the analytical procedures, what methods are you using to characterize your API and your drug product? And then, of course, how are you justifying your specifications? So you need to include all of that in there, and in order to be able to do that, you have to make your drug. Note that this is not the case for veterinary medicines.
I have one of my clients, again, thinking about creative strategies that people use. You don't actually have to make GMP material for vet products. And so one of my clients has a sarcoma, a dog sarcoma model. And so they're able to do the full product and get you see it, just astounding results from a vet med perspective. And so they're sort of doing their proof of concept in that way.